All right, ladies and gents. So today we're going to pick up with the topic of protein synthesis. And if we remember that word synthesis means to create something. So in this case, we're taking all of this information, these sets of instructions that are coded for in your DNA, and we're going to talk about how your cells actually use that DNA code to create these proteins, which is really the driving force behind how cells look, how they function, how you as an entire organism are capable of doing everything that you do all day. It's because your DNA has the set of instructions, but it's really those proteins that are actually doing all of the actual work. So, how does this work? Well, when we talk about your protein synthesis process, there really is this central theory in biology called the central dogma. And remember that theories are these scientific principles in a particular field of science, in our case biology, that are based on lots of evidence, and they're utilized as a way to explain the way something works. So in this particular situation, the central dogma is a theory that states how this information flows through our cells, and it only flows in one direction. Your DNA is the original set of information. The proteins are the ultimate product that we produce. But here's the caveat to this. When we look at DNA in a cell that's doing its job, that means we're in interphase. That means, at least in eukaryotic cells, that nucleus is in place. The DNA is trapped in the nucleus. But if you remember, the site of protein synthesis is your ribosomes, and those ribosomes are outside in your cytoplasm attached to the RepER or free floating. So, how do we get the set of instructions that are currently trapped in the nucleus out to the cytoplasm to actually build those proteins? Well, that means there needs to be an intermediary molecule, and that molecule that makes that gap or bridges that gap for us is RNA. More specifically, we'll talk about it as mRNA. So, when we look at the central dogma, it really is made up of three central processes in your cells. First and foremost, it's the DNA replication, and we've already talked about this. How do your cells continue to make more copies of that genetic information so that when it goes to make the new cells, that new cell also has that same set of instructions? That's where the DNA replication occurs. When your cells are just doing their normal everyday job, their job is to read their set of instructions, their set of DNA, and create the proteins that they are designed to produce. Well, we just stated DNA is stuck in your nucleus. This really is think of it as the master blueprints for every potential product that our cell could create. Now, if we're talking a muscle cell, that muscle cell is going to access certain parts of the DNA that tell that cell how to be a muscle cell. If I go to a neuron, it's going to access a different set of DNA instructions to be a neuron. Sort of think of it like going into a library, and you are a science major, so you're generally going to access the information that is related to science. But your roommate is a law major, so they're going in and accessing the part of the library that is designated for studies of law. Your friend down the hall is an English major, so they're going and accessing the library for just the English lit section. Well, that's kind of what we're dealing with. So DNA is the blueprints or the entire library. But you, as that particular cell, only access the pieces that you need. And that's where the RNA ultimately then translates into the product that you're building as those proteins. Another analogy that might work for you, think of DNA as the entire encyclopedia of recipes for cookies. Every cookie you could ever want, every recipe is housed there in that DNA. But today, your factory is only making chocolate chip cookies. That will ultimately be the protein product at the end. So, all you're going to do is access the information for how to make chocolate chip cookies today, and that's where that RNA molecule comes in between. So, going back to those three processes, you've got DNA replication to ensure that every new cell has the set of instructions that it needs. Once that cell is built and making use of those instructions, doing its job, that's where these processes of transcription and translation come in. So when we talk about transcription, transcription is to make a copy. If you've ever heard of a scribe in sort of ancient times, their job was to sit there and just copy documents before the printing presses existed. So transcription is just a copying process. Go back to the cookie analogy. So your DNA is every recipe you could ever want for cookies. Today we just want the chocolate chip cookie recipe. So going from DNA to RNA, we're going to transcribe or copy just that recipe that I need. Translation then comes into play when we take that set of RNA, that particular set of instructions out of the nucleus, go to those ribosomes, and actually create it. So, where does that term translation come in? Well, if you think about what it means to translate something, we usually talk about translation between languages. So, going from English to Spanish or English to German or something of that nature. What we're talking about here is translating between molecular or molecule languages. We're going from that DNA RNA world, which are your nucleic acids, if you remember your macromolecules, and we're translating that nucleic acid language into amino acid language, right? And amino acids are your building blocks for your proteins. So we're translating it between molecular languages. Now, here's the thing. When we look at this idea of the central dogma and taking that information from DNA to RNA to protein, we're really talking about this idea of gene to protein. So go back to the recipe idea. For every gene that is in your DNA, that gene is essentially the recipe. So you've got a gene for chocolate chip cookies, you've got a gene for snickerdoodles, you've got a gene for sugar cookies. That's what your DNA is divvied up into. It's divvied up into these chunks of information that are specifically designed to create a single product. So when we talk about how much of your DNA ultimately codes for a single product or a single protein, there's this whole idea out there of a single gene to a single polypeptide or a single protein. There are some exceptions to that rule, there's some tweaks to that rule when we're talking about eukaryotic cells. For any of you guys who eventually maybe look at AP Bio, we'll talk about it, but overarchingly, we're talking one gene to one protein, one recipe to make one type of cookie. Now, those proteins are your link between your genotype and your phenotype. So, if you remember what those terms mean, right? A genotype is your set of alleles. Do I have two capital A's, a capital A and a lowercase a, two lowercase a's? So, my homozygous dominant, heterozygous, homozygous recessive. So your proteins are that link between the actual alleles, which really are what's coded for in your DNA. When we talk about that capital A, it's its own set of A's, T's, C's, and G's. We talk about a lowercase a, it's a slightly different version of the same trait, the same gene, but the sequence of those A's, T's, C's, and G's is slightly different. And that's because when we look at your protein that gets produced from that, there's a reason that we have different phenotypes based on whether we've got two capital A's, two lowercase a's, or an A capital and a lowercase. So when we look at your phenotype, that's the physical expression of the genotype. Genotype is the code in your DNA. The phenotype really is the protein that we're actually producing. So if I have two capital A's, that means that my cell is producing two identical copies of the protein. And let's just say that particular dominant trait is purple. That means my cell is producing two copies of that purple protein. And so as a result, my flowers look purple. If I'm talking about my heterozygote, right, so they're the ones that have a capital and a lowercase, right, two different letters, then we're talking about being able to produce a dominant purple protein, but also a recessive white one. However, because that dominant is so strong, I'm looking at a purple flower anyway. I can't see the white protein even though it exists in my cells. If I talk about homozygous recessive, where I have two lowercase a's, now I'm talking about my cells producing two white proteins, and so my flower looks white. Hopefully that makes sense. If not, don't struggle with it too much here. We're going to come back to it when we talk about genetics in the next unit. But it's all kind of interlacing and interrelating to each other. So I want to try and make sure we take note of that while we're here.